that the most important people in your project team are actually the biologists and not the chemists. So, but like, I take that. So, it's okay. Okay, so I'd like to introduce Dr. Carolina Borsoy Moraes. She has a degree in biological sciences and PhD in microbiology and immunology from the Federal University of Sao Paulo. And in fact, she is currently a visiting professor in the Department of Pharmaceutical Sciences at the same university. And her research focuses on phenotypic screening for drug discovery, especially for neglected diseases such as Chagas disease, malaria, and leishmaniasis. So I'll hand over to you, Carolina. Have fun and enlighten us. Now, I think that you got to use this one.
Okay. Thank you, Peter. So, first of all, I want to thank Carlos and Andre. I think he's not here now, for the invitation to be here today and participate in this nice meeting you organize always. And it's very nice to have a chance to show to you some of the work we've been doing, and also in collaboration with Carlos group. So I will try to be very start from the beginning and explain to you how these essays works. And then if you have any questions, you can ask. But yeah, I am a biologist, and I'm more comfortable with cells, but I use compounds as tools. Yeah, I agree. Biology is very important in the drug discovery process, because that's where you get the insights to do something really new. Of course, without the chemistry, you cannot advance. So it's important that we talk and understand each other, so then we can move forward in a meaningful way. So Andre asked me to present a talk on bioassays for evaluating compound activity. And I'm working mostly on phenotypic screening, so I will present to you concepts behind the work we do using some real-life examples from our lab. So what is phenotypic screening? Is when you use cells, you use cells and evaluate the phenotype of the cell to measure compound activity. Okay, so you can go from very simple phenotype like viability to very complex phenotypes like when you see nuclear transport translocation into the nucleus from the cytoplasm, or from the membrane to the cytoplasm. You can design assays that will show to you that biological phenomenon, and we will allow you to quantify it, so you can measure how your compound or your compounds modulate that biological phenotype or phenomenon. Okay, and you can get very robust data from it. So you can use phenotypic screening for measuring activity of drugs, of chemical probes. So some compounds are not good for drugs, but they can become very nice tools for us to interrogate cell function, protein function. So these are the chemical probes. Antibodies also, so people working on immunologicals, you can use a phenotypic screening to measure antibody binding and activity in cell assays, and also gene function. Nowadays, there are many different techniques for measuring, doing genomics on cell-based assays that you can screen whole genome libraries for based on CRISPR activity or CRISPR substitution of individual genes, and you can find genes related to those phenotypes that you're looking for. So all these can be, you can use phenotypic screening for that. One branch of phenotypic screening that is the most useful one is high content screening, because it brings a lot of information. So this is One example, what basic things we can do in high content screening. So here you have two different cell lines, okay, human cell lines, U2S A549, and you can stain all the major organelles of these cells. So you can stain the nucleus, the ER, the nucleoli and the cytoplasmic RNA, the Golgi, actin also, which is not an organelle, it's just one structure in the cell, and the mitochondria. So you can see the different two kinds of cell have slightly different patterns of staining, right? And for each of these descriptors we call. So we do this, and we take pictures in the automated microscope that can take lots of pictures without help from us. So the computer does all the work. And then we take these images to software that can identify these structures. And then it can count the structures for us. So it saves a lot of time because usually when you do this kind of cell-based assay, you have to take a picture and analyze the images by yourself and count things or measure patterns. And then you have to compute that. So when you do high content screening, the computer does a lot of this work for you. And it's much more easy to, it's faster. And you can have very complex data, like based on several different kinds of phenotypes. And you can combine these to get the answer you want. Okay, so phenotypic screening is has several steps before you can get to the real experiment. You have to consider your objective, your goal in your question. What do you want to know? So even we work with computers and automated microscopes and so on, nothing of these will be useful for you if you don't have a clear question in your mind, okay? The computer does not make the questions for us. You cannot just come and say, I culture these cells and I want to see how they look. No, you have to know what you want to look, what kind of phenotype you want to see. Then you can start developing your assay. So you have to choose the kind of fluorescent marker you use, like you make a GFP protein of your protein of interest, or do you use dyes or antibodies labeled with dyes? And you have to measure, you have to estimate the kind of the size of your sample. So are you going to do high throughput screening and screen thousands and hundreds of thousands of compounds? Or are you going to test a small series of compounds, of analogs, and you want to measure just the difference in activity among them so that you can build your SAR? So this is very important because we'll determine the kind of workflow we have here. Okay, so obviously for very large compound libraries, we need a lot of automation to do the sample handling, and this is sometimes not available. So any academic settings, anyway, most people have handful of compounds, hundreds of compounds, or a few thousand of compounds. So usually we don't need a lot of automation here. Then we have to define the imaging conditions, and then analyze the data, okay? So we can get the numbers we need to interpret the phenotype that we want to see. Phenotypic screening has made kind of a comeback in recent years because for many years, target-based approaches have been the major line of investigation for biologists working in drug discovery. So this is basically when you have a target protein of interest that has been previously validated in a disease model, and you develop biochemical assays to measure activity on this kind of in this protein. I guess you have seen many examples over the days here these days, and you measure activity of compounds directly on the protein target. Okay, this kind of approach has many advantages, and I always hear many chemists saying it's much superior because it gives information on the structure of the target and binding mode of your compound to the target, so you can design compounds more easily based on this kind of approach. And this is true, it has this advantage, and you can know the mechanism of action with in a lot of detail, so you can know how the compound, the drug is binding to the target, and you can use these to improve your compound, your drug. And it is a very successful approach for a number of diseases, has been very successful. But it also has some disadvantages. And the target, and this is where the biology is really important, when you validate your target, there are many complications that can lead to false positives or results that are don't translate so well later in the clinic. So this can be a major cause of drug failure in development. So when you take your drug candidate to the clinic, it doesn't have efficacy. So why is that? Because in vitro, your drug binds to your target and has the activity you expect. So why the compound, the drug does not does not have the therapeutic effect you expect? Maybe sometimes, and many times, can be because the target somehow is not relevant in the disease. So target validation is really tricky. It really depends on how good your in vitro and in vivo models are for your disease. So this is not very easy to achieve, okay? So if your target is not really thoroughly validated, maybe you do a lot of work on that target, but it becomes meaningless years ahead, and you have spent a lot of time and money on that, and that's, of course, not what we want. And also, sometimes you have in vitro data that does not correlate well with in vivo data, which make it more difficult. So in recent years, like I said, phenotypic screening has made a comeback. Why comeback? Because in the beginning of history of pharmaceutical drug development, most drugs were tested in phenotypic approaches. So they would give compound to an animal or even directly sometimes to people and see if it works. And if it has efficacy, then you get a drug. That's how it used to be many, many, many years ago. And some of the drugs we still use these days, several of them have been discovered by this way. But of course, as science progresses and we become more analytical and we want to test molecular hypotheses, this became less and less used because it doesn't give you information on the molecular level. And it was not seen as good as this target-based approach. But with more technological development and also facing the failure of target-based approaches in some cases, phenotypic screening became again possible and viable because there were development of these automated microscopes, and now with artificial intelligence that you can use computers to analyze data for us, complex data that arise from this kind of screening. And it became, again, an interesting idea. And nowadays, most drug discovery programs, they use both approaches. It's not like one is best. It's just something is better for something. And sometimes, phenotypic screening is better than target-based. But if you have the target, you can also work on it directly. So it really, it's more useful to have a combination of both, okay? And one of the greatest advantage of phenotypic screening is that it's much easier to get a target that is relevant for the disease in your model because you will not pre-select that target. So you just look for efficacy of your compound, you see the activity the compound has, and then you go and try to find what the target is. So this process of finding the target is very laborious, and sometimes frustrating because not always you happen to discover exactly what the target is. But when you do, usually you find a new target. So you can have a chance of develop new therapeutic classes because you have new targets. And then it gives you great competitive advantage. So it can be very useful, especially in the case of infectious disease, because we just usually want to kill the pathogen. We don't care how the pathogen die. So in these cases, phenotypic screening is very, very interesting. And in case of neglect diseases that I will talk about to you, it's even more interesting because there are very, very few targets that have been validated. Even the validation is highly disputed among the people, the biologists working on this disease. So it's an interesting approach because you don't get a bias from the beginning and you have higher chance of success. And in fact, has been used by major screening centers working on these diseases these days. So when you look at the past literature, you can see, like I mentioned, nobody is just picking one kind of strategy. They use both to find new drugs because they are very complementary to each other. So when you think about infectious disease, you can see historically phenotypic screening, like I said, has been very successful in the discovery of new first-in-class molecular entities. And here you can see the comparison of the two types of approaches, phenotypic and target-based, and for small molecules. And you can see first-in-class usually are discovered when you use a phenotypic approach. But once you already know the target, and then you can develop best-in-class drugs. And for that, then target-based approaches are very, very useful because then you can really improve on the compound property, knowing the structure of the target. So it's important to keep this in mind because it's not useful to be talking which approach is best. You just have to know when one is more useful than the other. But even phenotypic screening has a lot of advantages for speeding up drug discovery and discovery of new drugs with new targets. It's not useful if you don't have a good assay. Okay, so of course, everything we biologists do in vitro is just an approximation of what happens in vivo in the patient. Okay, we cannot reproduce the complete complexity of a disease state in the tube or in the animal even sometimes. So it's very important to know how much your model approximate your disease, okay? Otherwise, you'll be pursuing targets that are not relevant in or will be not relevant for drug efficacy later on. So here just you see a model state, an in vitro model, for instance, and the disease state. They don't share many features, and the main pathways that drive disease development are different, that drive phenotype are different into these two kinds of models. And then what you see in your assay is different than what you see in your disease. So the more you approximate these two, the higher chances of success you have. And also these days, we have a lot of technology. This is progressing very fast, and we have capacity to work with more complex in vitro models of cells. So the more complex your model gets, more likely you are to have something that approximates your disease more, and of course has a better predictive value. But the complexity, of course, also increases the cost. So you have to consider how much you want to increase complexity, because otherwise it becomes unfeasible sometimes. So what do we recommend? Usually you have a screening cascade when you're doing phenotypic screening and supporting drug discovery programs for one disease. So you have a primary screening, which is a simple screening that will give you a more basic response. So for example, working with infectious diseases, you can look here for compounds that will just kill the pathogen, okay? You don't care how. You just look for activity or sometimes not cytotoxic activity, not cytoactivity, just static activity that will prevent pathogen to grow, okay? So this is really simple and easy to do and much cheaper than more complex assays. So you can do a lot of compounds in this kind of scheme. Then you select compounds that have the activity you desire. We call these compounds hits. And these hits, they have to be subject to improvement to reach properties that you expect for a drug, okay? So as you know, this process that we optimize hits to lead and then lead compounds that are being optimized by the chemists. And we, the biologists, provide support to give you biological data to guide this process, okay? So here we can do simpler assays, again, just to check for activity of your new analogs, or we can do more complex assays that will tell a bit more about how the compound is killing the pathogen, for instance, or what is the mechanism of action, cellular mechanism of action of those compounds. And this will help the team to prioritize the compounds that have more, that have a better profile and have more chances of successfully advancing for, preclinical research. So you can use phenotypic screening in all these steps here, and you can do also optimization based on the structure of the compound. So you find hits that have similar structure, so they are analogs, and then you can design compounds based on those structures, or you can find hits that have similar biological profiles. So they have similar activity on the cell, cause similar phenotypes, and sometimes they are not chemically related. So this is very interesting for us, for the biologists, but for the chemists, it can be challenging because how you will improve these activities if you have no clue what parts of the molecule are important for the activity. But both kind of approaches can be useful depending on your goals. So I'll just give you example of some collaborative projects we developed in recent years. So the first one, it was a consortia of several groups, international consortia, and NMA3PI, New Medicines for Trypanosomatidec Infections, where we developed a platform for screening libraries of compounds synthesized by the chemists of the consortium, or also purified from natural products that were obtained from microbial origin, and against T. cruzi, T. brucei, and Leishmania. Okay, so Chagas disease, human African trypanosomiasis, and leishmaniasis. So we did a phenotypic screening platform for this, developing assays for these 3 diseases. And then we screen our collection of compounds made inside the consortium against these 3 diseases. So this is just to give you an idea. This is a typical screening result. So each point here is one well in the well plate. So we work with several those microplates that have 384 wells. And here is the activity of the compound, okay? So in these assays, they were very simple assays with just looking for inhibition of parasite growth or infection. Okay, so brucei grows alone in the medium. It doesn't contain host cells, doesn't infect host cells. So they behave more like bacteria, just growing in the medium. And then we measure how these molecules inhibit their growth. You can see here, the yellow ones are the compounds in the library, and the blue and green and red are controls. Here is the green. And so here's positive and negative control. So usually in screening is very, very common. Most compounds don't have activity. They just follow normal distribution. So if you plot this activity, you have a normal distribution, the population here of compounds. But those that have activity, they differ from this pattern of distribution. They cluster together with the controls. So you can see here many compounds were active against Brucei, which is really common. So anyone working with Brucei out there will know that it's very easy to find active compounds against Brucei. So, and usually they are very, very active. So this parasite can be easily inhibited. Their growth can be easily inhibited. So you have a lot of hits. In this case, it was like 11% of our collection had more than 90% of activity, which is a lot. Okay, so you have to do more assays to select those compounds because there are far too many here to work with. So then we can select the really best ones to move forward. In case of Cruzai, which is also a trypanosoma, but it's very different from Brucei. It infects the host cell, mammalian host cell, and then we measure activity of the compounds against these intracellular forms of T. cruzi. So they grow inside the host cell, like the cells of our body. So if you know, people with Chagas disease have a enlargement of their hearts, for instance. So they infect heart tissue and they grow inside the hearts. So we can measure activity of these compounds that will inhibit the growth of cruzi. You can see it's already much more difficult to kill cruzi. You have hits with activity starting at 60% inhibition of growth. So a big drop in activity from this assay to this assay, which is just, not expected or unexpected. It's just to illustrate that two different parasites have very different behavior. Okay, so sometimes it's easier, sometimes it's more difficult. And in case of cruzi, we measure also activity against the host cell, so we can see cytotoxicity, which is another important and crucial point that we have to see how selective the compounds are. And in our case, we can do that in the same assay. So our hit rate dropped to 8%, which is less, but it's still a lot. So again, we need to prioritize these compounds to see which ones will move forward. In the case of leishmania, it's even more difficult to find compounds that kill leishmania. We don't know that why that happens exactly, but it has probably to do with the metabolism of the parasite, because leishmania grow very slowly inside host cells in our in vitro assays. And probably because they are not so metabolically active as the other two, there are less pathways that we can mess up with, and then they are more resistant. This is a pattern of activity that is becoming more and more clear for us working in parasitology. Parasites that don't grow much or don't reproduce much, they are more difficult to kill, okay? And so in case of leishmania, we find very, very few compounds here that have activity above 40%, which is not even good, okay? 40% is less than 50 is not good. But if we don't choose these, we have nothing to work with, so we work with what we get, and we get less good compounds here, but it's okay, we have to move on with this. So I just want to show to you that it really depends on the assay, the kind of parasite you want to target, but it can be really easy and you publish a lot, but maybe here, because it's so easy, there is not so much need here. And these two are more difficult, so there is still a need to find new drugs against these two. And you use these assays to screen this diversity library in this project, NMTRPI. So with synthetic compounds, the screening cascade is pretty simple. We do one primary screening and then select the hits. And these hits in this project, they were further screened in a target-based screen. So like I said, the combination of target and cell-based approaches. And then we found the hits that we want to move on for optimization, okay? And in this project, we also had a library of extracts and fractions, 11,000 extracts and fractions deriving from fungi, so microbial origin, okay? And these were both screened in parallel, in phenotypic screen and target-based screen. So here there were several targets the project had already selected for evaluating, because they were sort of validated before. So they want to work on those targets. And then we select the best hits based on the antiparasitic activity. So at this point, it doesn't matter have a target activity. If it does not is not active on the phenotypic screening, we drop that fraction, because there is no reason to prioritize that. Probably it will not have activity in the cell. So because it's fractions, we have to do several rounds of fractionation of those fractions to isolate the active compounds, and then test the purified compounds on our assays. So then we evaluate the structure of the compounds to see if they were new or if they are viable. Some compounds are not, they simply are too, they were not possible. We had no way to purifying them because they were very liable and they would degrade when we get to this point. And then we go to heat validation. So we did all this effort, and here is just for you to illustrate comparison. So here there were several kinds of compound classes the consortium was working with. And here we have phenotypic screening activity, and here target-based screening activity. So you can see sometimes some classes have great activity against the target, but very poor activity against the parasite, and others were good for both, and some were okay for the parasite, and also moderate for the target. So this is important to counter and screen in the phenotypic screening when you're using a target-based screening assay, so that you can know your compounds will actually have activity against your intended target, the pathogen. And now I will show to you some examples of more detailed studies we can do with phenotypic screening to not only analyze the profile of the compound, biological profile of the compound, but also to prioritize those compounds that have activity that we desire, and also to use compounds as probes, like I mentioned in the beginning, to study the phenomenon we want to know, not necessarily use them to develop drugs later on. So this is maybe... Okay, so this is a typical image we get from our Leishmania assay. So here is the nuclei of the THP1 macrophages. Okay, so these structures. And you can see, like here, faintly stained is the cytoplasm of the THP1 macrophages. And inside the cytoplasm, you have these tiny dots. These are the parasites. So they are phagocyted by the macrophages, and they start to grow inside the macrophage. Okay, this is how they infect human cells. So you can see they form these vacuoles which are full of parasites, and sometimes they are not in the vacuoles as well. So we have these two kind of structures. Anyway, cells are heavily infected. So we take these images and we develop the analysis protocol using the computer. Okay, I will not go into that kind of detail, but we already publish, and if someone has interest in doing this, it's all available, and you need to have access to high content screening system. But then we can measure, okay, not only activity, but also the biology of the parasite. So we can count parasites and see how different species of Leishmania, in this example, behave in the host cell. So we use two species, Amazonensis and Brasiliensis, that infect humans and cause cutaneous form of Leishmania, so skin infection. And we use two species that infect internal organs, so they cause visceral leishmaniasis, which is Donovania and Infantum. This is the most prevalent in Brazil, and these two also available in Brazil, exist in Brazil. And you can see the different species, they don't behave the same inside the macrophage, okay? So this is a simple assay, in vitro assay, but you can see there is a lot of biological variation here. And when you take a drug to the leishmaniasis patient, you will know which species is infecting, but we don't know more than that. We don't know how that species will behave, because this is not only difference between species, several different strains of the same species also behave differently. So it's can be very complex, because you have a lot of iteration between the genotype of the species, the genotype of the patient, which we also don't know how interplay with the disease. And then you have to take all this into consideration. So one practical way of taking this into account is making assays that will consider part of this diversity, putting this into your screening cascade. So you have an idea if your compound has a higher likelihood of having good activity against this variety of conditions, or if doesn't have a good activity profile, I guess these many species, maybe probably in the clinic, will not be as successful as we hope. Okay, so we develop assays for these different species. You can see, so they infect less, and they also have less parasites inside cells. So there's a lot more parasite here than here. Okay, and we validate these using reference drugs. So amphotericin B is the drug that we use to treat leishmaniasis. And you can see this is a good drug from the point of view of efficacy, okay? It's not a good drug considering other aspects, but it's a good drug because it has good efficacy in vitro and in vivo too. So you can see, despite all these differences, here is the dose response curve. So activity against the parasite is increasing here. So it means it's inhibiting infection. And here is drug concentration is increasing. So for all four species, you have a very similar drug curve of activity, which means that a good drug usually will have activity despite all the biological variation, okay? So this is one way to mitigate the impact of biological variation that your compound will face later on when it goes into clinical trials. So we use these assays to screen a small library of 100 compounds, the pathogen box, which is freely available from the Medicine from Malaria Ventures, to compare the activity of these compounds against the 4 species. Why we do that? Because then we can work from the start with a compound that has higher chance of being, of have broad spectrum activity against several leash species. So this is a 400 library, a collection of compounds. And you can see when you screen against these 4 species, of the 400, only 6 had desirable activity against the 4 species, okay? And this library is already clustered into, the compounds were regionally discovered against several diseases and in several disease drug discovery programs. So you can see in the case of leishmania, many compounds arise from TB programs, which is interesting because they infect the same host cell. So it gives you clues, you know, later on when you were to look for a mechanism of action, it's useful information. So shifting to Chagas disease now, this is typical image of our assay. So again, host cells, okay? These are U2S host cells. In this case, cell type doesn't matter much because T. cruzi can invade any nucleated mammalian cell. So they invade and multiply inside the cytoplasm. So heavily infected cells, and we put compounds on it and then measure compound activity. And we use the same library to measure infection activity against 3 kinds of strains. So cruzi is only 1 species, but has a lot of genetic variability within the species. So some strains, some type. There are currently 7 genotypes for cruzi, recognized by most people working in this field, and they are not so evenly distributed in Latin America. So here in Brazil, we have more of the genotypes 2 and 5 and 2 and 6, but especially 2, which is represented here by the Y strain. In the southern part of the continent of Latin America, we have mostly 6 and 5. So here, CL Brenner is group 6, and Silvio is X101 is a strain from the group 1, which is more common in other parts of Latin America. So Colombia, Mexico, and Central America it's more easily to find humans infected with this strain. Okay, the other genotypes are more difficult to work with, and they have been mostly isolated from wild animals. So for this study, we focus on these 3. Okay, so you can see they also behave differently in the host cell. And here we can already see some difference in the activity of benzimidazole, which is the main clinical drug for Chagas. And these 2 strains, Silvio and CL Brenner, were very, very susceptible to benzimidazole, and this Y strain was more tolerant to benzimidazole. So we have a shift in the activity. It means you need much more benzimidazole to kill this Y strain than the other 2, okay? So when you look at the compound activity here, so this is showing the differences between the 3 assays, the 3 screens, we can see again, out of 400, only 4 had good level of activity against the 3 strains. And then we can see again, TB was highly represented, but in this case, were compounds that were already previously described with activity against cruzi or brucei. So we could not find anything very new here. But anyway, just to highlight how difficult it is to find compounds have broad spectrum activity, and this kind of assay should be considered sooner or later when you're doing synthesizing and developing compounds for these diseases. Another topic that is very important in the case of Chagas disease is to consider these efficacy drugs, because we can have different kind of activities, and this is really crucial, something we learned in recent years. So this is hypothetical, okay? You're testing 2 drugs in a cell-based assay. Drug A has this curve in red, and it has high efficacy and is more potent, so it requires less drug to achieve 50% of inhibition, which is the EC50. And drug B is less efficacious, doesn't kill all your pathogens, and it has a higher EC50. But so in this case, it's really very easy to say drug A is better than drug E in this assay, okay? But in case of real life, sometimes we get this. Drug B is highly potent, so it has IC50 that is sometimes even nanomolar range, in the nanomolar range. And drug A is not so potent as drug B, but it's more efficacious. It can kill, reduce more the infection than drug B. Okay, so usually when we are looking at compound activity, we just report IC50 values. Okay, so if I just report to you IC50 values, you say drug B is the best one here. But when you look at the efficacy, then drug A is better. So which one should we consider? So we did this with benzimidazole and posaconazole. So back when we did this study, posaconazole was a drug that was in clinical trials for Chagas disease, okay? And it was a very promising drug because it was the first trial that happened for Chagas disease ever. So people were very hopeful it would result in a new drug to treat Chagas. And then we developed this phenotypic assay using different strains of T. cruzi, each one belonging to a different genotype. So highly diverse here, okay, genetically speaking. And we test benzimidazole and posaconazole. So benzimidazole, again, was similar against all strains. So variant in potency was variable, but it always reached efficacy sooner or later, okay? So it reduced infection to undetectable levels. On the other hand, posaconazole had very variable activity. So some strains, like this one in blue, which is group 5, is more common in Bolivia, infection in humans with this group, and is not really very much studied in terms of biology. So this group 5, or this group, this orange strain, which is commonly found in animals in the Amazon, and this also has been reported to be found in human patients. They were very tolerant and almost resistant to posaconazole. Okay, so posaconazole had almost no efficacy against these two strains. On the other hand, toluene, which is a strain most people work with in the lab, was highly susceptible to posaconazole, so it had high levels of activity, but not as high as benzimidazole. So you see here is almost reaching 100%, and posaconazole is around 80% here against toluene, and much less efficacious against the other strains. So we still didn't know which one was the best, but we know this one was highly variable when comparing different strains. So when we finished this work, at the same time, the clinical trial results came out, and this had posaconazole failed. So it had good control of T. cruzi infection while in therapy. So when the patients were getting the drugs, you could not find T. cruzi circulating in their bodies. But 1 year after the follow-up study, they found disease had receded, so they were showing infection again. And while benzimidazole doesn't have this problem, but the problem here is very toxic, so it's very hard to do this treatment. So actually, we saw that posaconazole was not as good drug as everybody expected and thought to be. And then we asked why? Why is that? It had such promising results. And then we realized because our assays to test posaconazole before the clinical trials were not the best assays that we could have. So we are only looking at EC50s and not efficacy in vivo and in vitro. So then we changed the assays, and now we look at efficacy as well, which is really important because it shows, it correlates much better with clinical data later on. Okay, so I'll go very quickly here. And like I said, T. cruzi can infect several different kinds of cells from different hosts, so all mammals, basically. And one question we have is how important is that for, for us, for disease or for even testing compounds? How different the activity of this compound will be depending on the type of cell that you're using as a host cell. So to see these, we test 4 different kinds of cells with the same strain of cruzi. And again, use a small collection of compounds that we purchase from the vendor just to measure the variability in compound activity in this, to see if we see the same variability in response when you change the host cell as we see when we change the parasite strain. So here are the images of these 4 types of cells. You can see some are more infected, like THP1 is heavily infected than others. Here the infection is not is much less. You can see many less infected cells here and here than here. But once they infect, they grow to dominate the cell. So we see difference in the biological profile of these infections. Some are more, and cells are more infected, and others are less infected. And same for the cell number. So depending on the host cell, for instance, Vero cells, even though they are not so heavily infected here, when they are infected, you lose a lot of cells compared to when they are not infected. So this just means for us working drug screening, it means that every assay have its special and specific features. You have to be aware of those when you studying or using these assays for screening. And you can see here we use benzimidazole again and ifertimox, which is another clinical drug for Chagas disease. And you can see, depending on the host cell, we have a very high activity of benzimidazole against this Y strain, or much lower activity. And ifertimox, less variation, maybe because it's just more toxic. So it's more like poison, actually. And this strain does not show much resistance, natural resistance to ifertimox. So depending on this, what this tells us, that depending on the tissue of the body that T-Cruz is growing, maybe if you develop a drug, it can have different activity depending on the organ, the target, and then you have reservoirs inside the body, okay? This is information. But anyway, again, for us working drug screening, this is important to know because we have to be aware of these biological differences so that we can test our drugs against these variables and see how it performs, okay? So we did the same exercise again, screening the compounds against all 4 cell types infected, and then number of compounds that had desirable activity varies depending on the kind of host cell. So THP1 is similar, as much as infected as U2OS cell, but for some reason we find many more hits in U2OS screening assay than in THP1 screening assay. So we selected the common hits that were active in all 4 assays, and then at least in 3 cell lines, and then we get 11 hits. So again, you lose a lot of compounds here, but it's normal. And this is just screening data. And then when you look at the 11 compounds, only 2 were active against all T-Cruz in all 4 host cell types. And there were some variability that we observed, but at this point, we don't know the meaning of this variation, but then we just focus on the most active ones. And here are some, sorry, it's a bit shift here, but you can see some compounds, again, were very active against the Y strain, which is the strain we did the screening on. And then we take other T-Cruz strains, you see the activity, the drop in activity. So this one is a C50 value, 1.6 micromolar against Y strain, and no activity against CL Brenner, okay? So same here, you see 0.4 micromolar against Y and 70 micromolar against CL Brenner. So this kind of variation is real and it should be considered. So we focus on the best ones that are in green here. I mean, sorry, this is not the best one. This is also a variation in activity, but this has interesting biological profile that we want to investigate further. So, and here as well, okay? So the best one we found in this screening is this, which is associated to inhibition of calcium metabolism. It's a experimental compound already developed for cancer, and it interferes with calcium metabolism. And we saw very interesting activity in the 3 strains. So around 2 micromolar EC50 values and high efficacy, near 100% efficacy. So we will be doing some studies on this compound. And how do we do that? So this is the last part of the talk. So like I mentioned at the beginning, you can use compounds as probes to study biological phenomena. In case of these parasites, they are very difficult to manipulate genetically. Okay, we maybe nobody here does this kind of thing, but when you try to transfect these parasites, delete genes, add genes, express heterologous proteins, they are not very easy. They don't comply with your goals. Okay, so you just sometimes cannot do that. Also, another interesting point is that when you have this situation, you can use compounds to modulate that protein of interest. So instead of deleting the gene, you use a compound that is active against the gene and study that effect on your biological system, okay? So another point is that when you do that, you approach the real situation you expect in the clinics. Because when you just delete a gene to prove that that gene is essential and then it should be a drug target, you're not deleting genes when you're treating people, okay? You're just modulating their protein activity. So when you do a drug, your drug will have to deal with the target present there and will have to shut off your target, but not remove your target from the cell. Okay, so in many cases, this is more relevant approach when you do this kind of interference than just deleting genes to prove they are essential. Okay, they are more relevant for real life situations. So, like I said, this is T. cruzi again, individuate cells. It has this flagellated form and it changes to a round form upon entering the cell. And this round form is called the amastigote, and they start to grow inside the cell until they fill up the cytoplasm, and then the cell bursts and they get released. Before that, they differentiate back in tripomastigotes and they infect other cells. So this is just to remind to you. Why I'm showing this to you? Because we don't know how this happens. I mean, how the parasite does this. We know nothing about that. What targets or proteins are involved in this process? We know very, very little about this. So how can we discover the proteins that are involved in this process? One way is to design assays that will show this progress, this process for us, so we can monitor how the parasite infects the cell, so we can monitor this stage and this stage, and then we can find compounds that will specifically inhibit some part of the process, okay? So this is what we did. So just to give you an idea, how we use a very simple screening assay to find this kind of specific compounds with this specific activity. Like I showed before, we have very highly infected cells and cells that have very few parasites inside. These are usually treated with benzimidazole, for instance, or active compound, and this is untreated cell. So if we want to find a compound that will modulate parasite infection, we want to find a compound that you have many cells with parasites inside. So you can see there are many more infected cells here than here, but inside the infected cells are very few numbers of parasites, okay? So here there are many parasites inside the whole cell, and here just a few. So this is what we were looking for, compounds that do not kill the parasite, but inhibit the progression of the infection. And we did this screening, not going into that detail, but we selected these in pink here, were the compounds that had the kind of activity we wanted. And we developed these then more sophisticated assay. We use markers to monitor the parasite. So this marker can stain in green the round stage, the mastigote, okay? When they differentiate to the flagellated state, the tripomastigote, they lose this marker. So we can know at which point of their development they are. And we look at this infection for several days using our marker, and you can see in normal situation, parasite invades the cells, and then after 24 hours, it starts to grow here. So here you have more green, more parasites compared to 24 hours before. And after 3 days of infection, you can see, if you look closely here, you see parasites in red, the nuclei of the parasites, but they start to lose their markers. So they are differentiated to flagellated tripomastigotes, okay? So this is normal infection cycle in vitro. When we do our screening, one of the compounds we selected, it had the ability to activity that would inhibit the progression. So you can see the parasites in green here in similar levels to the controls, But as the time goes, the cycle does not progress. So they stay inside the cell. Here you can see the green remains in very small numbers, okay, instead of here. And they, it's like they are frozen there, okay? So this was the phenotype we are interested in. And this is very specific because when you treat with benznidazole, you can see here, they start to lose the staining even early, like 12 hours of drug exposure in 12 hours of infection. So you have less and less parasites because they are dying. And when you reach the end of the infection, they disappear, but not because they change the shape of the cell, they differentiated just because they are dead. Okay, so this is a specific phenotype that we found using our assay developed exclusive for this type of investigation. And when you look the detail, when we want to profile the mechanism of action of this compound, we saw that it can inhibit DNA replication in the parasite. So in blue here, you have host cell nuclei, and the parasite are here. It's very tiny dots, but if you cannot see, believe me, they are all here. And we put this in the medium, we give them some uridine analogs that have fluorescent molecules attached to them. So the cell DNA will replicate, incorporate these molecules, and they become green, okay? So this is a replication assay. And when we put our compound together, we can see host cell continues to replicate. So they are incorporating uridines, modified uridines in their DNA. But the parasites, which is here in the arrow red, but I guess you cannot see on the projection here, they remain blue, so they are there, but they do not incorporate this modified analog, and they do not become green. It means they are not replicating their DNA. They have arrested at a point of the cell cycle where they don't grow anymore. Okay, so we can quantify this, and we can see this is specific. And why is this interesting for us? Well, biologically, there are many implications for this because nowadays we know, we are beginning to understand that non-dividing parasites are the cause that these diseases are basically incurable. Because even if you treat your patients, no drug existing now can get rid of these parasites from the human body. And this happens in vitro too. So if you give drug in vitro and you remove drug and follow up, they just grow again, okay? They remain there. These forms now are being called persisters. They exist also in bacteria, and apparently is a really, really relevant phenomenon that happens in microorganisms, but we know very little now about this. So everybody wants to understand how these persisters are formed. And we think one way is using this kind of approach, finding drugs or molecules that modulate the growth of these organisms without killing them. So then we can understand what are the targets of these drugs, and then know what proteins are involved in these processes, okay? So we can use the probes, the chemical probes, the molecules, as a way to study gene function and discover new genes. And also you can use this kind of screening assay for resistance studies. So if you have a target, you can ask your biologist collaborator to design proteins that are resistant to your compounds, because they can modify the active site for you, for instance. And then you can use the phenotypic screening to study these. So we have parasites that are resistant to posaconazole and ravuconazole, and we've been studying these using phenotypic screening. So I'll go briefly here. The resistant parasite, this one with R, is sensitive to benznidazole and nifurtimox, but has a high resistance to posaconazole. So here you have 17 micromolar EC50 for posaconazole, compared to the sensitive one when you have 2 nanomolar. So like 1,000 times increase in resistance to the drug. So this is also useful too, that if you are studying a compound, you want to know the target, you can ask your collaborator to select resistant cell lines, resistant parasites, resistant bacteria to your compound, and then you can measure resistance and use these resistant ones, you doing genetic studies to find out the target of your compound. Okay, this is it's a lot of work, but it's straightforward. Okay, so yeah, this is what I wanted to show to you today about phenotypic screening. Thanks to everybody in my group that was involved in these essays, these results I showed to you. Also thanks to Unifesp, where I am now, and in the Adema campus. A lot of this work was done in CB, in USP, and we have funding from the NDI, which is major organization funding Chagas disease and leishmaniasis drug discovery, and also Europharma for leishmaniasis project. Thank you.
Thanks, Carolina, for showing us something of the sort of power of phenotypic screening in drug discovery. I think we've got time for a couple of questions. I've got a very quick one then. I was just wondering about the sort of role of when you see the variation in efficacy between the different sort of host cells and stuff. Do you think there's a possibility that that might be caused by differences in transporter profiles?
Yeah, likely this is at least one of the factors. Several of these cell lines are tumoral cell lines derived from tumors and human tumors, and likely many of them expresses, ABC transporters, and they pump out drugs. And yeah, likely one of the reasons. But I think also has to do with metabolism. So THP1 is the cell, so it's a macrophage, and it has a very different redox metabolism than other cells because it's important for cell function. So to kill microbes, they secrete a lot of stuff that they have a different metabolism than other cells. So THP1 cells, they have more acidic vacuoles where the parasites grow, so they inhibit the vacuoles. The acidification is one of the process the parasite escape host response. And so because of these, likely many drugs are not active in the THP1. This is one of the reasons. So if the drug cannot be active in acidic compartment, likely will have no activity against leishmania, for instance. Actually, this is the explanation when you don't have activity in these assays, usually it's due because of this.
Good morning. Thank you. Question, do multi-drug transporters, do they play any part in this system, like as far as effluxing out the drug before it has enough time to affect the parasite? And how should we consider that?
Yes, there is little research still on this. For T-cruzi, natural resistance, what people have been called, which are strains that we just get from nature without selecting for resistance. They just naturally resistant to the drug. The only thing that has been shown up today is ABC transporters. So parasites upregulate these ABC transporters, and they are likely to also play a role. So yes, probably it's a mechanism of resistance, and it's very highly adaptable because it's not specific to the drug. So they can easily select that from the repertoire of things they can do to escape drug. This is one of the major.
Okay, if we have no further questions, can we thank Carolina again and the other speakers this morning as well? Yeah.